Hello everyone, it's Mimi here. BMS working in the NHS. And today's video will be about tissue selection, which is part of the IBMS specialist portfolio in cellular pathology. So, just a quick introduction to this video. It will be short and sweet, I promise. I will discuss rationale and aims, learning outcomes, indicative content, resources, submission of evidence, Q&A suggestions, some evidence ideas, reflective statement, and some helpful tips to make your portfolio better. With all that said, I hope you will enjoy the content of my video and I hope you will learn a lot. So let's dive into this submodule. Rationale and aims of this submodule outline the general information and expected knowledge and competence that needs to be achieved when this submodule is completed. These are relevant to each submodule. For this submodule, the expectations are that the candidate will be able to recognize, apply and appraise the importance of maintaining the link between the sample and its request form. Work in a safe manner during tissue transfer of Category A, minimizing tissue loss or sample mix-up. Understand the impact of poor orientation during tissue selection and its consequences to subsequent procedures. Understand the impact of pre-analytical errors relevant to tissue selection submodule. Provide examples of troubleshooting relevant to tissue selection submodule. The learning outcomes demonstrate candidates' knowledge, skills, and competence in this submodule. They also include demonstration of the understanding of scientific principles, laboratory procedures, quality assurance, and professional conduct. They allow candidate to work autonomously and solve problems independently. Let's have a look at the first learning outcome, which is for the candidate to discuss the importance of the use of identifiers on the request form and containers to match, in avoiding sample mix-up. Why is it important we check patient identifiers on the pot and the form? What information do we check? How many identifiers are needed to accept this for analysis? What happens when the patient identifiers do not match? What could be the consequence of sample mix-up? Consider accuracy, reliability, patient safety, clinical risk mitigation, key identifiers, consequences of poor sample labeling, operational efficiency, legal and regulatory compliance, best practice, and so on. The second learning outcome is for the candidate to demonstrate how your laboratory operates to maintain the link between sample, cassette, and request form, including labeling and storage methods. Include how specimens are logged on laboratory information management systems, LIMS, and/or tracking systems appropriate to your own laboratory. How is the link between sample, cassette, and form maintained throughout the laboratory? What labeling system is in place? How is sample entered onto the LIMS system and how can it be tracked throughout the laboratory? Consider sample, form, cassette and slide labeling, LIMS system used, tracking system and accessibility of data, stored samples pre- and post-dissection, blocks and slide labeling, safety measures throughout this process, information verification during dissection, audit trail, traceability, possible errors, and how they can be minimized. The third learning outcome is for the candidate to recognize the hazards associated with tissue selection and explain the appropriate actions to maintain safe working, including steps to take in the event of an incident. What are the possible hazards that are associated with tissue selection? How do you maintain safe working environment during tissue selection? Consider biological, chemical, and physical hazards, cross-contamination, safety measures like PPE, ventilation, training, risk assessment, cleanliness, possible incidents like sharps or splash injury, biological spillage or sample mix-up. Consider steps you would take to deal with those incidents. The fourth learning outcome is for the candidate to demonstrate how you would set up the dissecting workstation in an ergonomic manner ready for dissection. How would you set up dissection bench? Why is it important the working station is set up in an ergonomic manner? Consider equipment needed, position of equipment, ability to set up the chair and bench ergonomically, working zones, lighting, accessibility of sink, bin, and so on. The fifth learning outcome is for the candidate to demonstrate with examples from your practice the transfer of Category A specimens from pot to cassette relative to your own scope of practice, maintaining appropriate audit trail records. How do you perform transfer of Category A specimens in your laboratory? How is the audit trail maintained during the transfer? What actions are recorded during the transfer? Where is the information recorded? Consider checking patient identifiers, SOP, methodology, specimens with special instructions, sample description and orientation if needed, scribing and recording information for traceability, possible sources of error and how they can be prevented. The sixth learning outcome is for the candidate to discuss the implications of specimen contamination and missing tissue and demonstrate with examples measures taken in accordance with laboratory protocols at specimen transfer to avoid specimen contamination and tissue loss for subsequent procedures such as processing and microtomy. What could be the implications of specimen contamination or loss during tissue selection? What measures would you take to minimize them? How would you deal with specimen contamination or loss? Consider measures like cleanliness, careful approach, following SOPs, disposable tools, careful sample handling, one sample at a time, sift through instead of transfer, use of or specie wraps. Consider implications of contamination and tissue loss on subsequent procedures and reporting like diagnostic and therapeutic errors, patient safety, operational impact, ethical and legal consequences or reputation. The seventh learning outcome is for the candidate to explain and apply the importance of correct orientation of samples at specimen dissection and state methods taken to distinguish them in subsequent tissue preparation procedures such as embedding and microtomy. What is the importance of correct sample orientation during tissue selection? What impact does orientation have on subsequent procedures? What could be the implication of incorrectly orientated specimen? What sample types need to be orientated during tissue selection? How is the orientation need communicated for subsequent procedures? Consider sample types needing orientation like skin or tubular structures, reasons behind tissue orientation, ways of tissue orientation, ways of communicating orientation instructions to the embedders and cutters, use of ink, printing information on cassettes, instructions within tracking system, instructions on the request form. The eighth learning outcome is for the candidate to demonstrate with examples troubleshooting problems which may arise pre- and post-analytically, including root cause analysis. What are possible pre- and post-analytical problems that can affect tissue selection? How would you troubleshoot them? What implications could they have? Pre-analytical errors like specimen misidentification, incorrect sample labeling, mix-up of samples, sample sent dry, sample lost in transport. Consider post-analytical errors like delayed reporting, transcription error, poor result interpretation or misdiagnosis. Consider troubleshooting steps like error identification, source of error identification, corrective and preventive actions, communication, and so on. Those are the learning outcomes that have to be met for this submodule. Indicative content highlights knowledge and understanding requirements that might be assessed during the viva. They may or may not be demonstrated by the learning outcomes or candidate's evidence. In this submodule, candidate is expected to have knowledge and understanding relevant to sample contamination and how it can arise and how it can be avoided. How would you minimize sample contamination? Environment, equipment, and technique used during tissue selection. What equipment, technique, and environment do you use during tissue selection? Further or alternative methods of fixation that might be required for specific samples. Does your laboratory use alternative methods and why? Hemorrhagic, high-risk or tumor tissue samples. How would you deal with hemorrhagic, high-risk and tumor samples during tissue selection? Indicative content highlights skills the candidate is expected to be able to do. The following are: candidate can set dissection bench. Would you know how to set up a dissection bench? Can assist with dissection. Would you be able to assist during dissection? Can follow SOPs. Can you follow SOPs correctly? Can log errors. Can you log errors correctly? Can give macroscopic description. Can you give macroscopic description of specimens during tissue selection? Candidate must also be able to take appropriate actions when they are dealing with missing tissue during tissue selection that needs to be in accordance with laboratory protocols. How would you deal with missing tissue during tissue selection? Able to identify tissues requiring orientation. Do you know what tissue needs orientation and how it needs to be orientated? Able to communicate clearly the need for correct tissue orientation to embedders and cutters. How would you communicate orientation needs to embedders and cutters? Able to operate and maintain cassette labeling equipment. Can you operate and maintain cassette labeling equipment? Resources section on the Brightspace contains some very useful information. Submission of evidence on Brightspace informs the candidate what evidence needs to be submitted for each subsection. Candidate needs to submit answers to questions that were set by the training officer. I will share some examples of possible questions later on. Candidate also needs to select an additional piece of evidence relevant to this subsection. I will also share with you some ideas of possible evidence later on in this video. Each submodule requires the candidate to submit their reflective statement relevant to their practice and training in each submodule. Questions should be set by the training officer. They need to be based around the knowledge requirements, relevant for each subsection. They should allow the candidate to demonstrate their understanding in each subsection. The questions should be specific and relevant to the candidate's in-house practice. They should also allow the candidate to demonstrate their specialist knowledge. If you would like to see specific example questions, please have a look at the video for trainers. Link will be included in the video description. These are some ideas you could explore for your chosen piece of evidence. And some more ideas for your chosen piece of evidence. Each submodule also contains reflective statement which is asking you, the candidate, to summarize your role in the laboratory in the context with each submodule. How do you demonstrate that you can draw conclusion from what you have learned, reflect on your current practice and how it can change in the future due to gained knowledge? Think about what is your laboratory in context this submodule. What impacts can your actions have on your colleagues, department as a whole and for the patients? You also need to write a personal reflection on your training and why you chose your evidence. This is very personal to your own circumstance. It should demonstrate your ability to be self-aware when analyzing the gaps in knowledge. You can use it as an opportunity to reflect on aspects of your training, application of your gained knowledge and skills, and how you have achieved your goals. Think about what your evidence demonstrates. Why did you choose this specific evidence for this submodule? Also write what your training was like when it comes to this submodule and how your practice changed due to your training. The reflective statements can also be used as a CPD evidence in case you are chosen to be audited by the HCPC. I would also like to share with you some helpful tips that can make your portfolio look really good. Try to present the information in various formats such as tables, pictures, photographs, diagrams, flowcharts, graphs or even videos, if you can. This nicely breaks down a lot of text that is usually present in specialist portfolios. Try to make it a little bit more interesting to the reader, which will be your training officer and eventually the assessor. Try to put yourself in their shoes. How would you like to read hundreds of pages of plain text? There is a saying I really like, and it goes like this: a picture is worth 1,000 words. I will make a video. I will share with you my evidence I used for my portfolio back in the day. Hopefully that will help. Also try to use various types of evidence, not just one type, like case studies, for each submodule. You could use experiments, case studies, annotated results, annotated photographs of you performing tasks, evidence of troubleshooting, annotated EQA results, non-conformances, validation and verification evidence, comparisons of different approaches and their pros and cons, control material production, certificates of course attendance with reflective statements, annotated images or charts, and so on. Try to bring in your personality. Think about the ways you like to learn best. If you like experimenting, perform experiment. If you like troubleshooting, perform troubleshooting. Choose something that demonstrates your understanding, knowledge, skills, and how you apply this in your practice. And one more thing, keep the learning outcomes in mind when you selecting your evidence. Good evidence will tick off several learning outcomes. And this brings me to the end of today's video. Hopefully you now have a better understanding about the learning outcomes for this submodule. And I hope you also have a better idea what evidence you can choose to demonstrate your competence. And my final take-home message? Keep those learning outcomes in mind when answering questions and selecting your evidence. Bye for now.